Great. Thank you, Dr. Serene, for giving me the chance to come in over here and give a talk. I was actually calculating this is 20 years after 20 years I'm giving a talk in India. The last time I was a master's student, and so I like to have a back and forth. So if you have any questions in the middle of the talk, would absolutely welcome the back and forth on that. So thank you so much. It's a great pleasure to give a talk at an institute which I have seen built up because I'm a resident of Vasant Kunj, or I used to be. The title of my talk is Unravelling Novel Biology Driving the Maturation and Senility of the Brain in the Gut. Now, what do I mean when I say brain in the gut? I mean the enteric nervous system. So the enteric nervous system is the nervous system that resides solely in the gut. And that animation that you see over there is not an animation, it's the real enteric nervous system. So we have taken about a centimeter long piece of the small intestine from a mouse, and we have completely optically clarified it, and then we've done light sheet microscopy on it, and that gives you an idea of the complex intrinsic innervation, the complex network that this nervous system exists in. And you can actually see this again if I can. I can't, but that's fine. So if you see it from the outside, it looks like a concertina wire, chicken mesh kind of a thing. It completely makes this network, and there are two different networks that are running concentric across the entire length of the gut. The outside layer, the outside network is called the myenteric plexus. It contains the most number of neurons. The one that's on the inside is called submucosal plexus. And together, they actually have about 500 million neurons and about 2 billion glial cells in the gut. And together, they regulate almost all the functions of the gut, including the motility of the gut, digestion, absorption, secretion, immunity, and many more. So they're organized, as I said, into concentric networks, and dysfunctions of this brain in the gut, or the enteric nervous system, are associated with or cause very different and diverse gut motility dysfunctions and disorders. So I'm an enteric neurobiologist, and I'm giving a talk to. So I'm an enteric neurobiologist, and I'm giving a talk to hepatologists and biliary scientists. So why is it that I want you to care about the ENS? The first thing is gastrointestinal dysfunction is pervasive in patients presenting with and without primary dysfunctions of the gut. And that includes hepatobiliary disorders. It includes liver cirrhosis. It includes patients with altered liver metabolism, PBCs. And as Dr. Serene was just telling me, that it also includes patients with acute liver failure. Gut dysfunction, in turn, leads to nutritional deficits that exacerbate hepatic insufficiency. So you have gut dysfunction, you can't eat well, you can't have nutrition, it alters your metabolism, it gives you a second hit to your liver. However, in either kinds of diseases, whether it's a primary disease of the gut or it's secondary disease of the gut, the ENS, which regulates key metabolic functions, is negatively impacted. That we know, but what is the mechanism driving it is something that we don't understand. So this translates to a lack of pathological insights, leading to lack of diagnosis of these patients, and lack of meaningful molecular targets, leading to a lack of therapies. So we have no cure for these patients, and there's not even a race for a cure. So that's why I care about these patients, I care about these diseases, and I think that's why you should care about them as well. The second question is why studying maturation and aging of the ENS is important. So the first thing is the metabolism of mammals changes significantly post-birth and with age. The gut is the main highway for nutrients, and ENS is the main regulator for motility. So one who controls motility of the gut controls the biology of every visceral organ. So if the brain in the gut is altered, every single visceral organ, including the liver, is shot. So I don't know if you're as big a fan of Pixar movies as I am. I started becoming a bigger fan of it when my son was born. But we have spent a lot of time thinking about how our brains mature. If you've seen this movie Inside Out, it talks about how as we become mature, how we become adolescent, how our feelings change, how our thoughts change. And if you've seen the other movie, Coco, then we have spent a huge amount of time, and there are lots and lots of institutes that focus on how we become senile, how we age. So this is all about the brain, but we have absolutely little idea about how the gut matures and how it ages, especially from the neurological side. And our current understanding of ENS maturation and aging is very observational. It lacks mechanistic insights. And to give you an idea about this, here is a table from a review, where they talked about all the different changes that they saw in various animal models and in human patients. And there is no mechanistic basis to it. And there are always these things that there are some neurotransmitters that go up, there's some neurotransmitters that go down, some neuronal populations go up, and some others go down. But there is no clarity on what happens. So when we're talking about the ENS, when we're talking about the enteric nervous system, what is it that we mean? So the first thing is, let's talk about what the enteric nervous system is. We saw what it is, but in terms of development, the enteric nervous system, when the embryo is maturing, when the embryo is developing, comes from this place called the neural crest. And the neural crest, cells from the neural crest migrate all the way down into the developing gut, and then they seed it. It's almost like a colonization of the gut. And this colonization of the gut continues till the animal is born, just before that. And these are enteric neural crest derived cells. This is the mesenteric side, this is the anti-mesenteric side, and they start going from one end into the other. And the entire innervation and the entire colonization of the gut is complete right before we are born, right before mammals are born. A failure of this happens in some patients, and when that doesn't happen, it leads to diseases called Hirschsprung's disease, where patients are born without a functioning enteric nervous system in the distal part of their colon. This is me preaching to the choir. Most of you are doctors, so you would probably know this. So if you think about the basic biology of this, let's think about it in a much more simplistic term. So you have a cell that is migratory cell. It comes from the neural crest during embryogenesis. And as it migrates down into the gut, it becomes what is called as the enteric neural crest cell. And from there, it branches either into a neural precursor or into a glial precursor, so into a neuron or into a glial cell. So every neuron in the gut and every glial cell in the gut traces its lineage back to this migratory neural crest cell. So if you are able to label any of this enteric nervous system precursor cell at this stage, everything down here will be labeled. So it's almost like putting a label on things and saying what exactly comes from where. This is something that we call as lineage fate mapping. Now, we all talk about the importance of perseverance, the importance of scientific curiosity, but let me talk a little bit about the importance of serendipity. So in 2010, when I was a postdoc, we started figuring out that if you take this, and this is the neural crest as it invaginates, and these are the pre-migratory neural crest cells, and once they start migrating out, they give rise to the peripheral nervous system, including the enteric nervous system. So our idea was that we want to see what the enteric nervous system looks like. What does all the innervation in the viscera look like? So for that, we made a mouse model called the Wnt1 T tomato mouse model. So Wnt1 is a gene that is expressed right here in the neural crest cells. So by doing a lineage fate mapping, what we did was we labeled every single cell of the neural crest with a TD tomato, with a fluorescent protein called TD tomato. So if you shine a light on it, a black light on it, it glows red. And you can see it over here because all the melanocytes in our body also come from the neural crest cell. So if you put a black light on this mouse, you can see that the melanocytes start glowing red. And so does every other part of the neural crest derivative, which is the peripheral nervous system. So remember that cartoon that I showed you, which is if you label everything from the precursor side, then every single cell at the end should label red. So every neuron and every glial cell should be red. So when we looked at the mesenteric ganglia of these animals, red here is the Wnt1 label. So everything that is red in this ganglia is coming from the neural crest. We labeled it with antibodies against a glial protein, GFAP. And when we see this, you can easily see that all the glial cells, which are in green, are also red, suggesting that all the glia are coming from the neural crest. So that's great. That's exactly what we would expect. But when we looked at the neurons here, again in the same neural crest derived mouse model, when we use it with antibodies against HU, which is a pan-neuronal marker, we see that there are some neurons shown here in the red arrow that are coming from the neural crest because they are labeled with red and green together. But there are lots of other neurons which are green, are not labeled with red, suggesting that they're not coming from the neural crest. So this is an aberration. This is something that is going against the dogma of about 100 years. So this is something that we serendipitously looked at in 2010. And in 2011, there was a paper that came out from Vasilis Paknis's lab at the Crick Institute that showed exactly the same thing. So they used a different marker for the neural crest. But again, over there, they found that every neuron labeled here in white There's some neurons labeled here in white that are coming from the neural crest, but then there are others that are labeled here in red that are not coming from the neural crest. So suggesting that this is not something that we just saw and it was an artifact in our lab, but this was something that was shared outside of our lab in various other labs all around the world. Now the second thing that we did was to find what exactly is the percentage of these neurons. So the Wnt1 positive neurons that are known as the canonical neurons, the ones that we expect will be coming from the neural crest, are about 50% in the adults, which means that 50% of the neurons in the adult gut, we have no idea where they're coming from. This is going against about 100 years of dogma, which says that every single neuron in the enteric nervous system, in the peripheral nervous system, should be coming from the neural crest. We have no idea where these neurons are coming from. And this is not just in one line. We spent about 5 years developing various different mouse models and seeing that this was the same thing replicated over and over again. So no matter which animal model we looked at, it was exactly the same pattern. 50% of neurons in the gut were completely unaccounted for when it comes to their developmental origins. So our cartoon now changes from we know that these are coming from the neural crest to a large population, about half the neurons in the enteric nervous system, we have no idea where they're coming from. So these were the questions that we started with. What are these neurons? Where do they come from? When do they come in? And what do they do? So the next 5 years was spent in doldrums, where we started looking at every single mouse model that could be available to us, and nothing worked. So this is the importance of perseverance, where we spent about 5 years looking at each and every mouse model available under the sun to see where they're coming from, and nothing helped. Until we went to a cardiology lab right next door to us, and we asked them for a mesoderm-specific mouse model. Now remember your embryology, the ectoderm gives rise to the skin and the neurons, the mesoderm gives rise to heart, muscles, smooth muscles, skeletal muscles, and the endoderm gives rise to pancreas and liver. So when we looked at a mesoderm-specific mouse, which is something that is supposed to give rise to muscles, we started seeing that the smooth muscles are labeled red, as expected. But inside the myenteric ganglia, we see that some of the neurons labeled here in white arrows are also expressing red and green together. So this was the first evidence that we had that it could be quite possible that we are overturning dogma of about 100 years, suggesting that there is a large population of neurons coming from the mesoderm. Completely crazy. So we wanted to be sure about that. So what we did was we took adult markers of the mesoderm, a myosin marker, so myosin heavy chain here, the slow tonic myosin heavy chain, and we labeled it in the Wnt1 Cree system. So everything that here that you see in red is coming from the neural crest. Everything that you see here in green is coming from the mesoderm. So when we overlap it, we see that they are mutually exclusive. There are populations of neurons sitting inside the myenteric ganglia in red, and these neurons are not red, as you see labeled with the white arrows, and they are MHCST positive, so they are mesoderm derived, and they are not coming from the neural crest. When we quantified them, it's exactly the opposite of what we saw in the neural crest mouse. So it matches that this is the hidden population, this is the unknown population of neurons that we now know are coming from the mesoderm. And we completed the loop by showing this again in the mesoderm specific lineage weight mapping mouse, where the mesodermal markers, developmental markers, the mesoderm specific markers in the adults, and the neuronal markers, they are all co-labeled together. So here is the evidence that these neurons are definitively coming from the mesoderm, and hence we call these neurons mesoderm derived enteric neurons. So now we have two populations of neurons in the gut from the developmental point of view. There is a neural crest neuron which is called NENS, and there is a mesoderm neuron called MENS. Now we didn't stop there. We wanted to make sure that this was not something that we just saw as an artifact. We had a look at this through multiple different mouse models, and we made sure that they are absolutely coming from the mesoderm. They also don't behave like canonical neurons. So the other question of what exactly they do, how do they behave, we can actually look at this in a mouse model where SNAP25, which is a pan-neuronal marker, which is the target for Botox, is expressed by all neurons. If you look at SNAP25 expression in the myenteric plexus, then you can see that there is exclusivity, that SNAP25 is not expressed by the mesoderm-derived neurons, it's expressed by the canonical neural crest-derived neurons. Now, this is where we ran out of the conventional tools, and then we went into single cell RNA sequencing. So we did a lot of single cell RNA sequencing to make sure with our collaborators that we have multiple populations that we found, multiple genes that we found, that we could then validate. So here is a photograph that shows 20 different markers that we were able to validate that really establishes the mesodermal identity of these neurons. So this then becomes the tale of two lineages. So there are two different classes of neurons. There is a neural crest-derived enteric neuron. There are more nitrinergic neurons, which we showed. There are more cholinergic neurons. And the mesoderm-derived enteric neurons are completely different. They have completely different functions that are hitherto unknown. They do purinergic signaling. They do CGRP release. They release cytokines. They release hormones. They do immunomodulation, etc. So if there are any person over here who is well versed with curing motility disorders, you would know that there are no good drugs against dysmotility. And we think that one of the reasons is that all the drugs that we have been using are targeting this population of neurons. It's just the 50% of them. A large population of neurons, the other 50%, we have absolutely no drug targets against because we have no idea what they are. So this is the hidden yes, absolutely. So the question is, why is the mesodermal neurons not going to the brain if their lineage is that thing? So there are two things. The first thing is that the peripheral nervous system, especially the enteric nervous system, is much more primordial as compared to the brain. The brain, in evolutionary sense, came in much later in the evolution of mammals as compared to the enteric nervous system. My favorite way of explaining this to Americans is you can have US presidents without a brain, but not without a gut. So that's how important the enteric nervous system is. The second way of looking at this is that developmentally, the brain closes out much before. And after that, after the blood-brain barrier, it's very difficult for cells from the outside to come in. I'll show the relevance of that in a minute when I talk about when exactly these neurons are coming into the gut. But that's one thing that we see. But to your other point, there have been reports that in the cerebellum, people have talked a little bit about, well, there is a strange class of neurons. Where exactly are they coming from? We don't quite know. So we don't really know because this is uncharted territory. So anyway, I'm sorry, does that answer your question?
That answers a little bit, but there are different populations when the mesodermal, and they migrate differently.
Yes. So
one last question is this, that how do these enteric neurons communicate with each other when they're migrating? I didn't see those synaptic network which normally is found in the nervous system when they are in the nerve.
Yes. So the question is, how do they communicate with each other? It's as compared to motor neurons in the spinal cord, where they form neuromuscular junctions. There is no neuromuscular junction in the gut because the neurotransmitters are released outside into the smooth muscles. So there is no direct communication between the muscle and the neuron. When it comes to the topic of how do the neurons communicate with each other, that's an even bigger black hole. We do know that there are neurotransmitters and neurotransmitter receptors, but whether the interaction happens at the synaptic terminal, that there is a real presynaptic and there is a real postsynaptic neuron within the enteric nervous system, is something that we are not very clear about. We do know that there is a lot of presynaptic and postsynaptic junctions between extrinsic neurons that are coming in, the vagal neurons and the sacral neurons and the sympathetics, and the intrinsic neurons. So it's a big topic of research right now to figure out how the communication inside the gut happens. So our model then is that there is one nervous system, two lineages, and many functions. Right now we are not just thinking about the enteric nervous system as something that is a prokinetic, pro-motility thing, but there is a lot more that the gut does other than just regulating motility. The big question over here is this something that we only see in mice, or is this something that we also see in humans? So based upon all the identification of the various markers of the mesodermal neurons, we took resected gut tissues from humans, and when we immunostained it again with the same markers, we stained it with markers of hue, which is here in green, which is a pan-neuronal marker. This is something that you guys might know better than me, which is c-Met, which is the receptor for HGF. So c-Met is very specific to the mesodermal neurons. And we see this expressed exclusively by the MHCST positive mesodermal subset of neurons. So MET is something that we see very robustly expressed only by a very large subpopulation of mesodermal neurons, not by the neurocrest neurons. That's where the connection between the liver and the gut becomes more interesting. But anyway, let me go on and talk about when do these neurons come into the gut, and that might answer your question a little bit. So at about P10, P11, so at birth, everything in the gut is coming from the neural crest. We know that. It's done through multiple different labs. But if you take an animal that is about 10-day old, which is when it's just opening its mouth up to start nibbling on some solid food, the microbiota has just started colonizing to a large extent, the amount of food, milk intake has started going down a little bit. That's when we see that there is this population of neurons that's coming into the gut, which is green, which is Hue positive, but not red. So this is the first instance at about P10 when we start seeing the presence of these neurons. So what we did was then we charted in different mice what exactly is the proportion of these two different lineages according to the ages. And when we did that, we were very surprised that at about P10, P11, there are about 5% of neurons in the gut, just when the animal is, coming into its being. At P22, which is when the animal is weaning, the teenage years, as we call it, that's when the proportions go in within the space of 11 days from 5% to about 30%. This is the most rapid expansion of any neuronal population that I've noticed anywhere. And thereafter, they still keep on growing, and at about a mature adult age of P60, about a 2-month-old mouse, they reach about lineage parity. There are equal number of neural crest neurons, there are equal number of mesodermal neurons. And this was very interesting because you can see that this has different phases that are associated with it. The P11 age is when they start having solid food, the microbial colonization increases. P22 is when they have attained sexual maturation, and P60 is when they have adulthood. So what else is the gut during this maturation phase? If you look at the two different and extremely important neurotrophic factors in the gut, GDNF, glial-derived neurotrophic factor, which is the supporting factor for RET. RET is expressed by the neural crest, is crashing significantly during this phase. So you can have detectable amounts of GDNF at P10, but you have very, very little GDNF left as the animal matures. So the ligand for RET, which is the supporting factor for the neural crest lineage, is crashing at the same time when the proportions of the neural crest lineages are shrinking. And at the same time, HGF, your favorite neurotrophic factor, your favorite growth factor, is significantly increasing. So you have two different things now. You have the RET, which is labeling the neural crest lineage. The supporting growth factor for that is going down. HGF, which is the supporting factor for MET positive mesodermal neurons, that's going up. So you have a seesaw effect in terms of the growth factors that are there in the gut. And when you look at where MET and RET are in the enteric nervous system, as I mentioned, MET is in the mesodermal compartment. It's not there in the neural crest compartment. It's there in the mesodermal compartment. And RET at the same time is only in the neural crest compartment. So you have these two different growth factors, two different receptors, two different lineages. And then what we did was try to do a cute experiment and said, well, what happens if you give GDNF into a maturing animal? So when you give GDNF to a maturing animal, the total number of neurons in the gut remain exactly the same. There's no significant difference. But the mesodermal compartment of neurons absolutely gets arrested. There is no change to it. So more GDNF, if you keep increasing GDNF and if you sustain higher GDNF levels, the neural crest lineage is sustained. The mesodermal lineage arrests. It does not develop. The other side of the story is what happens if you give HGF. Again, no difference to the total number of neurons, but the proportions of the mesodermal lineage expands rapidly. So if you are able to manipulate the growth factors in these mice during the maturation phase, you can either make the mouse much more juvenile, or you can make the mouse or much more infantile, or you can make the mouse much more mature. So the push and pull of these two growth factors allows the push and pull of the maturation. Now, we understand now this is what's happening during maturation. Now the big question is, does it remain at the same level? Does it remain stable at 50-50 as all throughout the age? And the most interesting thing that we found was, as the animal ages, the neural crest lineage is almost completely lost. So this is a P17-month-old mouse. This is about 80-85-year-old human in terms of age. the neural crest compartment is almost completely lost. And almost all of the enteric nervous system is made up of neurons that are coming from the mesoderm. The glia are still coming from the neural crest. So the juvenile phase or the infantile phase is exactly the opposite of the senile phase.
And what's the meaning of this, like microbial population which has more diversity, young ages is driving something. As you became old, the neurocrest ones, diversity decreased, neurocrest ones disappeared. What does it do to the physiology at that particular moment? How is it altered?
That's the thank you for the segue. As the animal matures, as the animal ages, we are seeing that the transit time, the gut transit time, significantly increases. So the physiology of the gut is short; it completely goes away. So in this mouse model, where we again have this is about a 60-day-old young adult. This is about a 15-month-old aging mouse. You can see that the transit time of a dye, which we use for as a proxy of gut transit, is significantly reduced in the aging animals as compared to the young animals. So the physiology of the gut in terms of motility is significantly down. Your other question about the microbiota, the microbial diversity significantly changes exactly at the same time. The hormones that are released by the gut significantly change as well. So there are significant changes that happen to the physiology of the gut that are associated with the changes to the neuronal phenotypes. But to the other question, what happens? Can you revert it? So we have shown the data that shows that if you manipulate GDNF levels, you can manipulate the proportions of the two different lineages. So the question then we had was, if you give GDNF at this level, can you normalize motility? So we gave 10 doses of GDNF to these mice, and as compared to the saline injected aging mice, which showed about a very delayed transit time, the GDNF injected animals showed a complete normalization of their gut phenotype, of the gut physiology. And when we looked at the proportions of the two different lineages, that was also significantly different. So we were able to rejuvenate in some aspect the physiology of the enteric nervous system, both at the cellular level, at a lineage level, as well as at the total motility level. So we think that GDNF can actually be a very good therapeutic for normalizing motility by normalizing the intrinsic biology of the gut. So this leads us to the question of, are MENs, the mesodermal neurons, are they senescent cells? So this is the definition of a senescent cell, that they should be present in normal proportions. In younger animals, they should have dedicated functions. So let's go through them one by one. Yes, they are present in normal proportions in younger animals, as we just showed the data. Yes, they have dedicated functional roles at health. They release specific senescence-associated cytokines. We see some evidence of that. We still are doing much more research into that. They are increasing in accumulation with age. Their accumulation with age is associated with dysfunction, and if you targetedly remove these cells, that's associated with normalization of function. So this is the talk that I generally give to a very basic science audience that says, well, that's great, you're doing translational work, but you have to go to the next mile and say, this is what we are seeing in patients. So this is where we are generally doing things from mouse models to humans. So now let me talk a little bit about how we are trying to do it the other way with the same story. How are we trying to take human data into mouse models? So my hypothesis was that what's probably happening is that if you do a XY graph between chronological age of the animal and the biological age of the tissue, then the healthy has a very normal linear 45-degree slope, but disease, you actually have accelerated aging, much higher biological age at a much lower chronological age. Do we see evidence of that? So if you take patients who have obstructed defecation, or patients who just cannot pass motions, and if you look at their bulk RNA sequencing, you see that the expression of these two genes, RET and GDNF, very, very neural crest specific genes, is significantly going down. So we decided to put that to test, and we saw that in their larger data, all the genes that are associated with the neural crest lineage, they're all significantly reduced in obstructed defecation. Genes that are associated with the mesodermal lineage, they're all upregulated. So this is the way by which we are saying, well, what we are seeing possibly might be true. But we have to not just do observational science, we have to do translational science, we have to take it back into mouse model. So what we did was, we said, well, we know that RET and GDNF are going down in these animals. So what happens if you have a reduction in RET? So we used a mouse model where there is a significant reduction, not a knockout of RET. And if you look at the whole gut transit patterns, so proximal markers of motility, between 9 weeks of age and 16 weeks of age, the motility patterns of the wild type RET positive animals remains absolutely conserved. There is no significant change. But the RET heterozygous animals, where there is a dose dependent reduction in RET, those animals start becoming much more, they start exhibiting signs of much slower transit. So this is something that we expect. This is something that fits our hypothesis that if you have a reduction in RET, then you're going to have accelerated aging, accelerated reduction in gut physiology in the same amount of time. Now then the next question that comes in is what happens to the proportions of the two different lineages? And when we look at it, between 9 weeks of age and 16 weeks of age, when we start seeing reduced motility, we see that the proportions of the neurocrest lineages absolutely crashes between just 7 weeks. You should not have that, because at 7 weeks in this age, you should have absolute stability. And at the same time, the proportions of the mesodermal lineage between the wild type remain absolutely same, but between the RET heterozygous animals, they show a significant expansion, suggesting that it's not just a crash of the neurocrest lineage, it's also an increase of the mesodermal lineage. So again, in these mice models, which replicate exactly what we see in those patients with obstructive defecation, we see exactly something that fits our profile. So this is the model now that we have, which is that reduced RET signaling beyond the physiological range accelerates ENS aging in a subset of patients to cause dysmotility. And we think that possibly drugs don't work well because the neurons that we target are not the dominating population anymore. Most of the neurons that we have the drugs against, they're almost already lost in these patients. That's why these drugs don't work against these patients. And the disease-modifying cures first requires us to normalize lineage proportions before we do the neuromodulator therapy. So to summarize my talk, postnatal ENS is developmentally dynamic with the first report of a mesodermal derivation of a significant number of neurons. The mesodermal lineage of neurons is phenotypically different from the neurocrest lineage. Both the lineages are regulated by their own trophic signaling systems. Lineage proportions are a biomarker of age, and caution is therefore warranted before we start thinking about cell transplantation therapies. So, more to the point, our work provides a framework by which we can understand biology of healthy aging, and this work should be published in about 10 days in eLife now. It also provides a mechanistic basis by which adult ENS is altered in disease. Senility of the gut is quantifiable. It's not just the brain that is where the senility is quantifiable now. We can actually look at the tissue. We can say what exactly are the proportions of the two different lineages, and we can quantify whether the ENS is mature, juvenile, or senile. And loss of one lineage is compensated by this. There are adaptive changes that we do see, but these adaptive changes are associated with alteration in physiology. Okay. So, food for thought, if you can reverse aging in the ENS, can that cause normative changes to the CNS through the gut-brain axis? Not just to the CNS, but to the other tissues as well. That's one thing that keeps me up at night. How are transmissible neuropathologies such as Parkinson's or tau generated in the aging ENS? And what happens to ENS in patients with PBCs, hepatic insufficiency, fetal alcohol spectrum disorders, where there is known gastrointestinal motility? We have absolutely no clue, but we think that our studies and our work establishes a platform through which quite a lot of these questions can be addressed, can be asked, whether this is associated with changes in aging patterns, whether it's associated with the loss of one particular lineage or the other. These are the things that we see. I was mentioning to Dr. Sarin that we see quite a lot of evidence that mitochondrial antibodies that you see prevalent in patients with PBCs selectively target certain specific lineages of neurons in the gut. That might be something that might be going on. Would love to work with you guys on this. But the last thought for the students over here, don't be afraid of challenging dogma. This is a quote from Jeffrey Bonstock, who was the father of purinergic signaling. And please go through this. This is something that is extremely important. If you are confident about your data, go where the data goes. Don't worry about what people say about it. I spent 13 years on this project. After 13 years, this project is now coming to a fruition. It's going to get published. It takes time, but persevere. Patience is the biggest virtue that you will find in this field. Thank you.